Hi, everybody. Welcome. You are in the first webinar of our new series, Microbiomes for All. This is sponsored by the ASM Education Department, and the facilitators are all from REMNet, and I'm sure that they'll tell you more about REMNet as we go along. So I'm Rachel Horak, and I'll be your tech support for today and your host. So if you have any questions for our speakers today, just ask them in the chat. And if you wish to ask anonymously, then I can bring that question to the speakers on your behalf. Just send me a private message in the chat and I will do so. If you need live transcripts, there is a button on the bottom of your Zoom toolbar and you can request live transcripts. If you need assistance at any time with tech support of using our Zoom platform, please send me a message through the chat. I'm more than willing to help you out. There'll be lots of times today where your participation will be strongly encouraged. So do take the time to interact with our questions, and all of our activities, because we're here to learn. And if you have any questions at all, feel free to reach out to our facilitators. They're more than willing to help you out. With that, I'm going to turn it over to our facilitators. And don't forget that this is being recorded and this video will be posted on our course LMS within about a day if you wish to watch it again.
Okay, are you turning it over to me?
I'm turning it over to you. I'm going to spotlight you.
Okay. Hi, everyone. Thanks for coming out this afternoon. My name's Theodore, and I'm from REMNet. And I just wanted to take a second to thank Rachel and Irene for helping us at ASM for helping us get this set up and organized. And also to thank all the people at REMNet who have been working on this, Davida, Avram, and Gina, who are here with us, and several other members of the REMNet community have been very helpful. And like Rachel was saying, if you have questions or things you want to interject, just shout them out, raise your hand, put them in the chat. That's fine. It's an hour. It'll probably go by pretty quickly, but we have basically a video of what we're doing, so you can kind of go back and check it. We have notes, and the whole idea is the REMNet community is here to kind of help you through this. So don't worry about not getting everything right from this webinar. There are going to be, plenty of other ways that you can get the information. And like Rachel said, we're happy to, just email us and we'll do whatever we can to help. So I'm going to share my screen. And let's see here. Can everyone see the screen there?
Yeah, you're good.
Great. So this is the first session on data analysis, and I'll just start by giving a little bit of background on REMNet. This is the Research Experiences and Microbiome Network. It's an RCN-UBE. That's a research coordination network for undergraduate biology education that's funded by the NSF. And the goal behind REMNet is to kind of work with faculty to help them get microbiome research into the curriculum. So that's kind of the basic gist of what we're out there to do. And obviously, if you're here, you probably already have a pretty good understanding of what microbiomes are. And you're probably excited about microbiomes like we are and like many of our students are. So I won't spend a whole lot of time on this other than to say that we really think that they're a fantastic teaching tool, that it's, very relevant to many fields of research. You can bring in all sorts of different courses, from intro bio to genetics to microbiome to statistics to ecology. So we really like that ability to kind of hit so many different fields with microbiomes. And again, we also just think they're pretty cool. So we recently put out a paper that I'm just kind of taking a picture of one of the figures here. If you're interested in kind of seeing what have people done with microbiomes in a kind of a classroom-based scenario or a course-based research. We put this probably a little bit out of date now, but we put together a list of references that kind of show publications where you can see examples of people doing this with Winogradsky columns and agricultural soils versus pristine soils and all sorts of different other microbiome examples. And so I really, encourage you, we'll send you the paper if you don't have access to it. I think the reference is down here in the lower left-hand corner. And this, might help you kind of see what other people are doing. It also has lots of techniques and protocols and so forth. And then, just the one thing that I'll say as a note on these microbiome projects as you're thinking about them is it's always good to have replication in your samples. It's always good to have some controls. Always good if you can set it up so you're kind of asking a question and testing a hypothesis. And what we're also going to spend a minute doing is taking a look at some of what our REMNet colleagues are doing. We have a couple short videos, and I'm going to turn it over to Davida, who's going to run those videos for you.
No problem. I'm just moving over to my screen now. I hope you all can see this. Let me know if you can see this, and most importantly.
I am Alice Taron, and I'm an assistant professor at St. Lawrence University. I teach undergraduate students in general biology, and I also conduct research with students. My interest is on soil microbiome analysis, particularly in their application to sustainable agriculture and bioremediation. I have conducted projects looking at the ability of wetlands to clean up groundwater contaminated with heavy metals and petroleum, and how a series of wetlands and aquifers are able to remove these contaminants before they are released to water. So I am interested in how microorganisms in the wetlands may be responsible for this cleanup of contaminated water.
And then our second one is.
Hi, my name is Christine Gertain, and I'm the director of a 3-year authentic science research course at Toms River High School North and Toms River High School South in Toms River, New Jersey. Our ASR students do DNA barcoding projects looking for the presence of a bacterium called Wolbachia, which is found in arthropods. DNA was extracted from mosquitoes in New Jersey and in Israel to search for the presence of Wolbachia in partnership with the Bordenstein Lab at Vanderbilt University. Students looked to schedule meetings across time zones, worked with partners that had English as a second language, and they learned that Israeli students attend school Sunday to Thursday. It was a true global experience. I also had 2 students work on soil microbiome projects. One was aquatic soil samples. The other project investigated the effects of insecticides on microbes in soil samples while they were growing ryegrass. The students used DNA subway to analyze their metabarcoding results. 3 sisters did research working on different types of oral bacteria related to tooth cavities. They each investigated different molecules that would inhibit bacterial growth, measuring zones of inhibition. In January, I will be hosting a bioprospecting training course that will train and certify teachers to run a bioprospecting research lab in their high school where students could learn how to discover novel antimicrobial candidates.
And then our last is Carlos.
My name is Carlos C. Galler, and I'm an associate teaching professor at North Carolina State University in Raleigh, North Carolina. I teach undergraduates and graduate students molecular biology techniques and skills, including bioinformatics and wet lab skills. And in 2013, we developed a metagenomics class. And we started by sampling kitchen sinks, and then we went across the campus sampling odd environments like downspouts, drains, water fountains to learn about the microbial communities that are around us. Our research question is, what interesting microbes are around us that we often overlook? What features do they have? What genetic potential do they hide that we can take full advantage of? And I use several different pipelines with students, including QIIME2, Nephele, and KBase. So far, we found some exciting microbes and some connections between the environment and our built environment, and microbes coming back and forth. Thank you. Take care.
Back to Theo.
All right, can everyone hear me again? Okay, great. So you can just see from that, there's a sample of, some really exciting projects, and, there are plenty more out there. So feel free to kind of follow up with us with questions or thoughts on what you're interested in trying out. The other thing that I think is really powerful about these microbiome projects is that they really give you a chance to target learning objectives that you have for your courses, as well as targeting many aspects of kind of quantitative aspects of biology and data analysis and statistics. And so this is another figure from that same paper. It kind of gives an example of, if you start at the top and go down, the different steps of a microbiome project and how they align with different areas of the microbiology curriculum. I'm a microbiologist, so that's usually the goals, the learning goals that I'm trying to target. But you can also do really, nice alignments with genetics or ecology or statistics and so forth. And so, the one thing we're going to be focusing on during the webinar here is at the bottom with the data processing and the data analysis and visualization. And the one thing that I would kind of encourage you to do is to be strategic in how you're thinking about this, because, what happens to me all the time is I jump in, I get really excited, and I'm like, oh, I got to do this, I'm going to try this and that. And to kind of help you focus and keep things manageable, if you're targeting specific learning objectives for your courses or for your students, that kind of helps you pick and choose, what types of analyses that you want to do, what types of data you want to collect, because you probably won't be able to do everything. It's just too much. So this kind of helps focus things. So today is the part one, and we're going to kind of get through the kind of nuts and bolts of how you start with your sequence data files, which are usually in FASTQ format, and get these loaded into the pipeline in Nephele that allows you to get data out of that. Next week, we'll go to actually looking at doing kind of more of the fun stuff and looking at alpha diversity, beta diversity, and some other measurements of diversity, and looking at community structure and so forth. And then we'll continue with even kind of more analyses in part three. But today is kind of the foundational day of how you kind of get things started. And the way that I'm kind of viewing this is that, we'll start with the microbiome sequence data. We're going to use these tools today. We'll be talking mostly about Nephele, but we'll also work with Microbiome DB and QIIME2 view. And with those excellent resources, we're going to get to the data analysis. And so that's kind of the mini formula that we envision. And we think this is the perfect time for a webinar like this, because these programs have, only in the last year or two become more user-friendly in terms of not requiring you to have to do any coding or do any command line stuff, which is fun in its own sense, but can sometimes be a hurdle for people that don't have a background there, including myself. So we'll hop right in and look at the microbiome analysis pipeline. This is kind of the big picture overview, where you're starting with your complex community. You're amplifying the target gene that you're interested. Today, the demo files that I'm using, they're real soil samples that I collected from around Brooklyn last December. And so you can have an amplification step that you do with students, or you can just take the DNA that you've extracted and send it straight to the sequencing company. sequencing facility, or if you have a sequencer yourself, you can do your own sequencing. We'll get all this data back. And then today, like I said, we're going to be focusing on the analysis part of things. And just put in this slide, you're probably wondering why am I spotlighting Repo Man, which happens to be one of my favorite movies. But whenever I think of Repo Man, I think about cars, and I think of some of the data analysis having to do with cars being, I don't know much about cars. I don't know what's going on under the hood, but I can certainly get from point A to point B. And so many of these steps, involve all sorts of, complex math, complex optics, complex computer science. But you don't have to teach all of those nitty-gritty details to your students. You don't have to have a fine-grained understanding of all of those various steps to really get a lot out of these tools. And so I'll just, remind you that at steps things will be, a little bit potentially overwhelming. But remember that, there are all sorts of experts that come together to put this information or to make these tools usable, and you don't necessarily have to be an expert at every step along the way. And so questions that we, I think in the prompting email that went out a couple of days ago that we asked you to consider. First of all, like where to sequence, I mentioned that a minute ago. We have used Wright Labs a lot and Mr. DNA a lot. I do a lot of Sanger sequencing with GeneWiz. A lot of you may have access to institutional sequencing centers, or like I said, have your own sequencers. But there's a lot of places now that'll do microbiome sequencing. We're going to be using Illumina data today, but I know that there are people out there that have Oxford Nanopore data, or maybe even Ion Torrent data. There are, a couple of tricks to kind of working with those data. But for right now, we're focusing on Illumina, and we're working with other people to try to get the tools that will allow people to work with, say, Oxford Nanopore data as well. Today, we're working on 16S sequencing from bacteria, but you can just as easily target ITS or 18S genes. You can do whole shotgun targeting sequencing. So you want to think about, what approach you're using. But today we're doing amplicon sequencing with 16S, since that's tends to be where people get started, and a lot of work is done with this amplicon sequencing. How much sequence do you need? Number of samples, the depth of sequencing. Typically, if you send samples out, the sequencing depth will be in the range of, say, 15 to 30,000 sequences per sample. Sometimes it's a lot more than that. And again, like I mentioned a moment ago, you want to consider if you're using mock communities or negative controls as part of your samples. And finally, if you're just interested in the data analysis and you don't have an interest or don't have the ability to kind of collect samples and extract the DNA, not a problem. We have data sets that we're happy to share with you. All of the data that I'm working with today is in the folder that Davida was talking about, so you can work with those FASTQ files. And also Microbiome DB has all sorts of data already in place that you can just do analysis with. So, oh my God, I just turned off the leaf blowers. I don't know if people could hear that in the background, but anyways. So anyways, it doesn't matter if you don't have access to sequencing, don't have access to data, you can get data in plenty of places. So let me just, I'm gonna pause just for a second to make sure everyone's with me. If there are any questions that popped up. Avram, was there anything in the chat that I should know about? I think you're muted if you're talking to me.
There was really just one question about mini-ion, mini-sec. Can I use my mini-sec for microbiome?
So this is the MinION, the Oxford Nanopore. The answer is yes. And I think Carlos and Davida, you guys have got some experience with that.
So we were answering it in the chat because the MinION data currently cannot be analyzed with Nephele, but we've asked for the folks at Nephele to start looking into that, because many people are using MinION. They have their own pipeline that you can use. MiniSeq, yes, I believe so. I've not used it, but any affordable sequencer, I have an iSeq. MySeq and all the other platforms work too. It's just they're more expensive. But you can pay for people to do it. And even the Ion S5, remember the Ion Torrent, people are still using the Ion Torrent platforms as well for this.
Yeah, so this is kind of a rapidly changing field, and we have been talking to the administrators of Nephele about getting systems set up so people can work with their MinION data. In any case, just to kind of provide a little bit of background on what we're doing here, you're going to start with these FASTQ files, which is the sequence data, or if you might be familiar with FASTA files, that was the format that was probably a few years old. The idea is that you're sequencing across this insert, which is a fragment that you've amplified. In this case, it's going to be a fragment from the V4, V5 region of the 16S gene. And you have Illumina primers that allow you to sequence in both directions across this gray insert. And so if you do single end reads, that means you're just using the data from read 1, which is what we're going to do today. If you're doing paired ends, it's going to merge read 1 with read 2, which is from the other direction. And so you get a longer sequence. And in just in case you're curious, the FASTQ format, it has your sequence. Well, first of all, it has a sequence ID. It has the actual letter sequence as well. And then this set of characters down here is a reference to the quality of the read at each base pair. So that's actually what's in that FASTQ file if you were to go in and look at it. And when you get the data back, this is a file that I get from the company that's doing the sequencing for us. And you're going to get a whole list of files that have your sample name embedded in there. It tells you that it's FASTQ. The .gz in this case means that it's compressed, but you can work with just FASTQ alone or FASTQ.gz. The Nephele works fine with either. And you can see that it says it's R1, which is read 1. That was the forward read I was talking about, and R2, which is the reverse read. And you can see that the sample ID for those is the same. The only thing that's different is R1 and R2. And so if you were to do paired end readings, it's going to combine those two so you get just one read out from that R1 and R2. Okay, and I'm going to turn it over to Davida for a second for the Jamboard.
All right, folks, so I'm putting in the chat, I'm putting the Jamboard link. So if you can all navigate over to Jamboard, I will give you a very quick tutorial on how to do this. So we wouldn't be ASM if we weren't having you all do some work for us, okay? So what we're going to ask you all to do is this prompt. What samples are you working with? And if you've not used Jamboard before, you simply go over, I hope you can see my cursor, I'm moving over towards, do you see that little post-it right there? I'm going to click on that sticky, and then when the sticky pops up, you simply write what you're working on there, and then it'll enter it and it'll appear on our screen. And then we can move everything around. So work away, whatever samples you're working on. If you're working on soil, poop, water, whatever it is, you can put it underneath there. But if it's something else, let us know. We'd love to hear from you. Yay. It's magic. Oh, there are too many people. Okay, so if you can't get in right now, and maybe there may be a limit on how many can go in at any one time. If you can't get in right now, I'll remind you all again in a few minutes. You can get in there. We'll reorganize this. And for those of you who are thinking about curriculum guidelines, Jamboard is a great technique to use in your class. It's very simple. Students really like it. Wonderful. Surfaces, paintings, masterworks. Wow. Some really cool stuff going on here. Okay. So moving the soils together. Lots of us working on soil. Theo will be excited. That's what Theo works on soil too. Okay. Not so many on poop though. I'm pretty disappointed. I wanted to see more poop. Wonderful. Or whomever wrote nothing yet, I hope you're coming up with some ideas already. Fermented foods and beverages. Holy water. Wow. Cool. Chicken respiratory tract. So there's loads of very diverse answers here, Theo. Lots to think about. I will tidy this up and I'll show it to you later on. I'll just reorganize it a little bit.
So Davida, are they going to be able to continue on this in the background if you switch it back over to me, or?
Yeah, I can keep working away so we can come back to it later.
Okay. So if you stop your share, then I'll, oops, no, where's my share? Okay, so hopefully everyone can see the presentation again. All right, so you've got these FASTQ files, your data. Now the question is, you got to analyze it. The trickiest, or well, I would say one of the trickiest steps in this is getting a metadata file that works. And so probably if you talked about these types of projects before, you're familiar with a metadata file. You also might have heard of a mapping file. The mapping file usually tells the pipeline where your samples are, where your data is, where it is on your computer, where it is on the cloud. And the metadata is obviously the data about your samples. And for Nephele, the kind of both of those things are combined into one. It's just the metadata file that you need. And on the right-hand screen here, you can see this is the sample. If you go to Nephele and you download the sample, a metadata file, this is what it gives you, and it tells you what it's expecting. On the left is one of the metadata files that I created. The key things are the sample ID. So you have to have the first column as a sample ID. The second column has to be the forward sequence. If you're doing paired ends, the third column would be the reverse sequence. And then in the case of single end, the third column has to be something called treatment group. And different pipelines have kind of different requirements in terms of, you want to have as much information as possible. And so I've got a different kind of version of the identifiers. I have short names, I have the source, latitude, longitude, date, collection site, all sorts of different things. I can't really think of anything, other than, if there's privacy issues, I can't think of anything that you wouldn't potentially include in metadata, because you don't necessarily have to use it. You can put in, the weather for that day. It may be that you never actually kind of use that in any of your analyses, but if you've got it there, that's great. So, but all of that can go in the metadata file after the third column or after the fourth column, if you're doing paired end. Okay, so I'm gonna go now from the PowerPoint to the Nephele site, and hopefully their connection is going smoothly. And I'm gonna try to run through this live on Nephele. If it doesn't work, I have slides as a backup, but let's see if I can get to Nephele. Okay. So can everyone see the Nephele window here? Okay. So this, if you just Google Nephele microbiome or Nephele NIAID, you can get here very easily. And this is kind of the entry page. If you look over on the right-hand corner, you can see that I'm signed in. If you have an account, you can sign in. If you don't have an account, you can create one. It's free, so there's no issues with that. And just to kind of get you oriented, there are three major branches of the Nephele website. So there's the pre-processing pipelines, there's the analysis pipelines, And then there's the explore pipelines, which kind of are an add-on to the analysis pipelines. You'll see up here at the top that we're at the home site. They have a workspace now. Here's my workspace, it saves your jobs up to 90 days, which is kind of nice. It used to be you have to kind of keep track of everything through your emails, but they have now this workspace. There's an excellent user guide that tells you about how to use Nephele. There are tutorials that are very useful, and there's an FAQ page. So there's really a lot of excellent support built right into here. And like I said, we've been working with the administrators at Nephele, and they've been pretty responsive. When I hit a glitch this weekend when I was doing something, I emailed them, and in less than 24 hours they'd fixed it. So I have lots of good things to say about Nephele. So the first thing I'm going to do is pre-processing. You need to think about this a little bit, because depending on where your data is coming from, you may or may not need to do this pre-processing. For example, from Wright Labs, my DNA comes with all the primers already trimmed off. It's already done some quality filtering. So I don't actually need to do the pre-processing with a lot of my samples, because the sequencing company has already done that. But you may have data that you're generating yourself, and you do need to do the pre-processing, or you just want to go through the process to make sure that everything is the way that you want it. The one thing that I'll point out again here is that you don't need to do this part with your students. I teach a microbiology lab. This is not one of the learning objectives for the microbiology lab. So I'm doing this on my own to make sure everything's in good shape. But if I was teaching a bioinformatics course or a data analysis course, I might do this step with my students. But again, these are the decisions that you should be thinking about: is I need to do this, or I want to do this, but do I need to have it be a part of the classroom activity? So anyways, you can learn more by just clicking on the Learn More button. We'll tell you more about the pre-processing pipeline. But what we'll do here is go to Start Job. And just so you know, this is pretty much how all the pipelines work. It kind of goes through the same steps. First thing it's going to ask you to do is if you're doing paired end or single end sequences, and we're doing single end for here. And then you do Next. And it's going to ask you to find your data. So this is kind of where the mapping file would come in, but you're doing it right here. You can upload files from your computer. I'm going to use a Google Drive. There are other options, but these are the two that I've worked with, and they work pretty easily. Especially if you're working with students, if you can put it in a Google Drive, then everyone can have access. And so in my lab, I do that so that students can get to the samples. Let's see, hopefully this is going to. Right. Okay, so Davida was yelling at me yesterday about how many files I've got here, folders, but I'm just going to find so this is my folder for this webinar. Here's my soil microbiome data files. And then here's the folder with my raw sequences. And so here you can see all of the sequences. Like I showed you, these are the FASTQ files. One thing that I'll point out, which I actually didn't realize at first, is that you can have other files that you're not using. So you can see here that I do actually have reverse reads for these samples, but the reverse reads were low quality, so I didn't end up using them. And that's a whole nother story I can tell you about. But initially I thought I needed to just have the R1 reads in the folder that I was going to take them from, but that's not the case, because if your mapping file specifies the file name, it's going to just, Nephele is just going to ignore any other files that are in it. So I'm fine with just hitting continue here, because these are the files that's going to pull out. And then, as I mentioned, I'm going to now select my mapping file. And the mapping file you've already created, okay, like I showed you a moment ago. And let me just see here. meant to have all this open in advance, but I guess I closed it down incidentally.
And TR, do you want me to drop in with some of the questions?
Yeah, sure.
Okay, so one question is, does each student need their own account?
That's an excellent question. And I think that, my students have their own accounts, but I don't, I think that you would probably, I think you're going to have, it's going to prompt you to create an account if you're, a student and you try to go in. I just told my students to go to the website and start working with it. So I think that they probably had to create accounts, but
Plus, they might overlap with one another if they were all using the same account, right?
possibly. I think, Carlos, is Carlos on right now or is he? Okay. Carlos has done this in larger classes, and so he might have some insight into that. But it's a good question. I haven't run into any issues with it, and I think the students are savvy enough to be able to, create their own accounts. It
How
might
about
be.
using,
Go
sorry,
ahead.
go ahead. How about using older FASTA files?
Yeah, so that was a question that came up, and I was talking with Carlos about that, and I was, we didn't have a great solution. I did find a site online where if you can do a little command line work, you can convert FASTA to FASTQ. But basically what it's going to do is just throw in some dummy quality values. And Carlos and I weren't sure that that would actually be a good idea to do. We've talked with Nephele about FASTA versus FASTQ, and we can try to get more kind of guidance from them on that. But because the FASTA doesn't have the quality scores built into it, there's you've got to find some way to get that information into from FASTA to FASTQ. So there's a cheat way to do it, and we're not sure that that's the best way to do it. But again, as we figure these things out, we'll circle back to the community or people that asked and let you know what we learned. So I'm in this process where I'm going to select my mapping file, and I'm actually going to start with one that has an error just so you can see what happens when things don't work perfectly. So this is my bad demo file, and I chose it, and now I'm going to upload it. Oh, why isn't it bad? Oh, that was interesting. I somehow. Okay, so let me just. Let's see, why did that? Huh, interesting. Okay, I'm gonna just show you what was bad about it, if I can do that.
Ina, as a graduate student, is recommending that students should have their own accounts, and she's given some personal account of what it was like when she did it. So that's in the chat. Thanks, Gina.
Okay, can people see my spreadsheet? Yeah, so this is what the bad spreadsheet looked like. And this has caught me a couple of times. Any column has to have some differences in that column. So if you can see in column D, everything just says bad. And one of the criteria, unless I just changed it in the last 24 hours, is that it's going to throw an error if everything in that column is the same. So if you have a column that's like, say, date, and you collected everything on the same date, that can create a problem because it doesn't want to see all the same date in any one column. So it's, just making you guys aware of that because sometimes that does come up. Other things that come up is sometimes the sample IDs don't match. Obviously, the syntax has to be perfect. So if you're using periods or underscores, that has to be match what the actual file is. So you can see that my sample IDs use periods, but the actual file names have underscores in them. And so that's another type of thing that'll throw an error. But anyways, I'm not sure why it's not coming up as an error right now. That's kind of weird. I just did it yesterday and it did pop up as an error, but today it's not finding an error. So you upload your file. And when it works well, which it is apparently right now, this is the next screen you get. And these are the selections that you're going to get in this particular QC pipeline. So remember, I'm doing quality control. This is just looking at the quality of the sequences. You can look at details about this process if you kind of follow that link to more information. It talks to you about what's going on in the pre-processing pipeline. Generally, you can stick with the defaults and that's fine. So I'm going to hit select, and I'm going to just put in a job detail, and you can see you have options here, so that if you did need to do adapter or primer trimming, you would do it here, and you could make the selections on what you wanted to trim off the 5' end or what you wanted to trim off the 3' end. And then if you were going to do quality trimming in terms of the quality of the reads that you wanted, or the quality at the front end of the read, or the quality of the back end of the read, you can make those changes here as well. But I'm going to just stick with the defaults. I'm just showing you where you can adjust those options if you want. And once you're fine with that, you can view selections. It's going to tell you what you've chosen. And then you can just hit validate and submit. Oops, did I hit validate and submit? Let me see.
Is it complaining about the options? Maybe you clicked the options and you didn't unclick it.
Oh, maybe, yeah. Okay, that's probably what I did. Sorry.
It's all right.
There we go. So once you have done the validate and submit, then you're going to get this screen, and it tells you what the job number is. And you're going to get an email and whatever email address you have associated with the account. And it's going to basically tell you the same thing. And so depending on the size of your data, this could take a few minutes to a few hours to run. And that's fine. That kind of depends on a couple different variables.
So,
And then you'll get a second email that tells you that your job has been done, or if you're checking in on your workspace, you get a notification there that your job is done, and you can go and look at the QC data. So let me just stop sharing this for a second.
there are two questions here.
Yeah, go ahead.
Okay, so why create an identifier samples column that essentially matches the sample ID?
Oh, that's not necessary. This was a project that had students collecting samples that use slightly different identifiers. So I just had an extra column, and I just put it in, but that you wouldn't, that's just kind of redundant. You don't need that. There's no requirement for that.
Okay, and are there any limitations on file size or number of users on Nephele?
I haven't run into any issues with file size. I think when you look at some of the pipelines, it tells you that there's a limit here. And I've had times where I've done the same run, and sometimes my data came back in like 15 minutes, and sometimes it took overnight. And I am guessing that has something to do with how heavily it's being used at any given time. But there is a way to check the status of your job. And that might reflect how heavily it's being used, but I haven't gone to look at that frequently. So the folder that's going to come back, and this is, again, all of these outputs are in the shared drive, so you can go ahead and look at them, is going to look like this folder here. And it's going to have a log file that you can look at that tells you what exactly happened, and if there was an error, that will show up in the log file. Importantly, if you actually did if you did trim things, or if you did do some quality filtering, all of your data, all of those trimmed reads are going to be put in this trimmed sequence folder. And so then for your next step, when you go to analysis, you want to make sure that you take these sequences, not the ones that you originally uploaded, or else you'll have just kind of bypassed all the QC you just did. But if you didn't do any, if you were just kind of playing around and you weren't really doing any QC, or you weren't really trimming anything, or weren't filtering anything, you can go back and use the original samples, or the original data. This was and so that QC report that came up there as a file, that's an HTML file, and it's going to take you to the multi-QC report that looks like this. And you can see over here in the last column, the percent dropped because I didn't put any filtering criteria on there. It didn't drop anything. And it gives you basic data on your samples. The thing that I think is interesting to look at here is the number of sequences. So this is the number of sequences in each of your samples. So you see I've got, several in the 30,000 range, but then I have a couple ones that are where I got very few reads, only 1,000 or 7,000. So it shows you that. There's lots of other information here. The other one I'm going to focus in on is these quality scores. So you can see that here is the position in the read, and we're going out to about 250 base pairs. And the Fred score, which is the type of quality reading that we're looking at, all of those Fred scores are pretty much all in the green region. And you usually want that Fred score to be above 25 or 27 to have pretty high confidence in the read. So again, there's a lot of other stuff if you're really into it to look at, but that looking at this graph and this first table is usually kind of what I'm interested in looking at. Okay. Right. Okay. So that's the QC. And what I'll show you next is, again, normally you would be waiting for your QC data to come back, but because we've already done it and I showed you the files of output, I'm going to jump straight to analyze. So if you've got your FASTQ files and are all set to go, you're going to just jump to analyze. And there are a couple options here. We're doing amplicon metagenomics, and there are a couple options for that. Mother, Data 2, Chime 2, and then there's the older version of Chime 1. What I typically use either Data 2 or Chime 2, and actually because it's all free and pretty easy, I normally use both Data 2 and Chime 2. They have a lot of overlap. The outputs are a little bit different, so that's why you might want to use them both, because I find there's some stuff from Data 2 I like, and there's some stuff from Chime 2 I like, and I use them both. But again, you're just going to you're going to click on that start job, and hopefully everything looks familiar here. You click sequence single end FASTQ files. You go and tell it where your files are. And back to my messy folder. Find my raw data. And then I just hit continue. Choose my mapping file again. I'll choose the good one this time, even though the bad one worked fine last time either. Maybe this one will get an error. And so here is what's going to happen in the Data 2 pipeline. There's a whole lot of processing steps. You can see that there's actually some filtering and trimming that's a part of the Data 2 as well. And I'm just going to stick with these default selections. And again, here you can see if you wanted to do some pre-processing, you can do that here. If you wanted to specify certain things about the analysis, you can do that here. We're going to stick with the defaults, which is the RDP taxonomic assignment database and the reference database of Silva. You have options if you wanted to change that, but we are going to just stick with these defaults. Job details. Again, I can put in a little name that tells me what I'm doing here. And then I can again view my selections to see what I've chosen to do. and then validate and submit. And again, same thing. You get a job identifier, and you're going to get an email that says that your job has been submitted. It'll give you a link so you can track the progress. And then again, in a few minutes or a few hours, you'll get another email that tells you that your job was complete. And that that's pretty much all there is to it. The one thing I'll say is that I'm not going to do it here just because I want to leave a few minutes if there's questions. It's the same thing for QIIME 2, exactly the same. It's going to look exactly the same. There's the same interface and so forth. So once you've got those basics down for the QC pipeline, for the Data 2 pipeline, for the QIIME 2 pipeline, it's all the same. And okay. And what I have in the slides, and I think we'll make these slides available to everyone, is just kind of still images of each of those steps that I just showed you. Here you can see when you get, I told you I only did the forward reads because the reverse reads were bad, and here's the evidence that the reverse reads are bad, so that's why I'm not using them here. But that's one of the reasons you might want to do QC, is to kind of see that. I think that Avram's been monitoring the chat, so this has probably been a work in progress. Avram, are there other things that came up in the chat that I should try to address?
Yes, would you use the QC data at this step or the raw data?
Oh, okay, good. Thanks for reminding me again. Let me see if I can find the, well, the answer is I would use the QC data if I'd actually filtered it. Yeah, so that's important. It's easy to kind of get distracted and just go back to your original files. Let me just, I can just show you again very quickly. So here's a folder with my QC, my post QC files. So I would actually select on those, and these are my actual filtered ones. And I would just hit continue here. The only thing to make sure that you're paying attention of is that, like I was telling you, the difference between using like periods or underscores, the output data for the QC sometimes switches your file names to have periods instead of underscores. So then you've got to fix your metadata file to match that. But that's not too big a deal, and it'll be pretty obvious if you hit that error. Other questions in the chat, Avram?
Annie Moore says Nephele is awesome for Illumina, but just curious, have the Nephele people said that they actually will work on accommodating Oxford nanopore data, or would those be, those of us using mini-ion look for something different?
What I can say is that when I talked to the people at Nephele, they're aware that there are people out in the community that would like to do it with Oxford nanopore, and so that they told me they were putting that on their list of things to get to. Now I don't know how big a priority that is for them, or if there are hurdles, technological hurdles, but we definitely are aware of those issues, and we're trying to find, if we find other solutions, or if other people in the REMNet community find other solutions, then we're going to do our best to kind of incorporate that into the support that we provide, and we're actively looking for that. But right now, with Nephele, the most success, or the only success that we've had, is using the FASTQ files. These other slides, again, this is what it looks like when you get a, when your mapping file fails. It's just going to highlight in red, and you can go in and fix it, either there, fix it there, or fix it back in your metadata spreadsheet. The one thing, so when you get your data back from Data 2, so here is the output from Data 2. The fun stuff, which will start in the next webinar, is all here in the graphs and so forth. But this is, again, the output file that'll give you a log file so you can see what happened, if anything went wrong. And again, some just basic data files that we'll talk about in the next session. And so again, the goal of today was to get you from the FASTQ into Nephele. And hopefully we were able to kind of show you all those steps. It's really, you know, I've done it myself, which means it must be easy. And my students have had a lot of success with it. So what I would say between now and next week is, if you have your own FASTQ files, try to get at least this far with Nephele with your own files. If you don't have your own FASTQ files, use our demo soil ones. And just kind of give it a shot, because it's possible that people will come back next week and say, well, I tried this and I hit this error that you didn't talk about, or what have you, and we'll try to address that. But according to my stopwatch, we're at 1 o'clock, and I just want to make sure that, you know, I know people have busy schedules, so I don't want to go over too much if possible. Any kind of wrap-up questions for the first session?
So we have a bunch of questions in the chat. Some of them are related to the DNA isolation protocols. So what we'll do is we'll pull those questions and we'll try and answer them offline, because we are running out of time.
Right.
But what we'd like you all to do, if you can, is that for next week, if you can at least try what we've described, and if you have any problems in advance of next week's webinar, we'll set up a Google Doc. You can ask your questions there, and we'll try and address them before we meet next week.
Yeah. So let me just, you know, like I said at the beginning, we really want to focus on the data analysis in this webinar, but we have all sorts of resources for, you know, what kits to use, how to use those kits, and kind of troubleshooting. We use mostly kind of QIAGEN Power Soil kits, but there are other similar kits available. If you have questions about kind of those earlier steps in experimental design and how to collect your DNA and where to send it, yeah, feel free to come back to us with that. We have lots of experience doing that as well.
And just one final call out to Alice. Alice is planning to have like a faculty mentoring network around MinION and data analysis specifically. So we'll be following up with Alice. And again, we hear you. Many folks want to use Nephele for just that. So I think if enough of us get in touch with them, they'll have to do it. Stacey is asking for a list of kits and where to send the samples. So Theo has been sending samples to various companies. Do you want to give a call out to some of the companies that you've used, Theo?
We use Wright Labs out of central Pennsylvania. And for REMNet, if you say you're affiliated with REMNet, I think they're doing $50 a sample, which is, I mean, you might be able to do a little bit better than that at a internal kind of house sequencing center, but that's pretty good. So that's Wright Labs out of Huntington, PA. And we also use Mr. DNA out of Shallow Water, Texas. And they're also, both these companies are very supportive of us, and they've been very helpful. Like I said, I use GeneWiz for some other sequencing, but you know, I haven't tried to explore all the options that are out there.
And there are very many kits. So generally speaking, we use a couple of different brands of kits, and we can put a list of things together for you. Can you say we're affiliated, or do we need to get affiliated? You're all affiliated now. Having participated in our workshop, now, whether you want to or not, you're on our mailing list, and you are part of REMNet. So we'll be putting you on the members list, and we hope to get you all involved in further events.
I'm gonna drop out now, but I think if Rachel keeps it open for a minute or two, Avram and David and Gina can answer questions. But I look forward to seeing you guys next week, and hit us up with any questions in the meantime.
Bye. Thanks, Theo. We are going to put everything in the Google Drive. So what you have there for session 1 will be stuff from session 1. We'll have a similar folder for session 2, and we'll try and put everything in there. But Rachel, as she said, she has a learning management system file already set up for this, so she'll be putting materials there as well.
That's right. So I just posted the Google Drive link that you all looked at today into your course LMS, and that's the same way that you originally entered today. Where you took your pre-webinar survey and you entered this webinar first. And so all of your resources that they're sharing will be stored in that LMS.
And
Go
one
if there's
final
any
thing.
more questions.
One final thing about REMNet, we have a number of working groups specifically, and one of our working groups is on access, availability of data and everything else. And another working group is specifically on isolation of DNA and protocols. So for those of you who are focused on that, and we'll be able to kind of ferry you through the appropriate working group, you can join them and work specifically on your problem of interest. So there's something for everyone in REMNet. We're a pretty inclusive group, we hope.
Thanks, folks. Okay, with that, Davida, I think I'll close down the room. Thank you
Nice
so
to
much
meet you all.
for coming. I apologize for the goof. It will not happen again.
We'll splice them together and no one will know.
Yeah.
All right.
All right. Take care, everybody.